Great, thank you.
Thank you.
Great. Well, thank you so much for the kind introduction and the invitation to come and speak today. It's a great honor to come and get to tell you about some of the things that we're doing to try to help advance microscopy, both in its spatial dimension and its temporal dimension. My day job is studying the brain, and in particular building tools that help brain scientists understand and repair the brain. But I think the themes that we'll talk about today are universal in biology. Throughout biology, we deal with differences in spatial scale, from molecule to cell, to tissue, to organ, to entire organism. And we have the issue of time as well. Diseases, treatments of diseases, aging, development take long periods of time, but of course, the elementary building blocks of biological computations are very, very short. So today I want to tell you 3 stories about technologies we're developing that are helping biologists cross space and time. And thinking about scale from the get-go, I hope you'll see, is really part of how we operate. You know, we have a very interdisciplinary group. Some might even say omnidisciplinary. Physicists and chemists, computer scientists and material scientists, biologists and clinicians. And the goal is really to bring together all these different fields together in the service of seeing and controlling biological systems with light. Now, spatial and temporal scales are challenging. If you think about the ways that we see and control biological systems with light, there are lots of examples over history where a technology can be built that works over a certain spatial scale or temporal scale, but it's very difficult to apply to a wide variety of biological questions. And that's why, of course, conferences like this exist. Innovation continues to this day in every regard. So the first half of the talk, I'll talk about our attempts to cross spatial scales to make it easy to image from molecule all the way to entire system. That'll be the first half of the talk. And the last half of the talk, I'll try to focus on some newer things, but we're really trying to help people cross temporal scales, and in particular to image many things at once in a living system. So let's start with imaging from molecule to system. Now, of course, many pioneers have developed fantastic ways of imaging with nanoscale precision. Recent Nobel Prizes have gone to inventors of super-resolution microscopy, cryo-electron microscopy, and of course, many other methods for nanoimaging have been developed over the last century, which have yielded huge numbers of great discoveries in biology and in medicine. But all these techniques require specialized hardware. They can be difficult for people to use or sometimes expensive. And all of them struggle to image large 3-dimensional objects. For example, if you're doing cryo-EM or super-resolution microscopy, it's difficult to apply that to a large intact block of tissue without processing further, which can be laborious and complicated. So in our group, we've been working on a technique that we call expansion microscopy. Rather than zooming in with some kind of lensing effect, can we take a biological specimen, preserved of course, this won't work on a living thing, and make it physically larger? So in our group, we often try to do the opposite of what people are doing, and this is sort of one of the ideas that came up through such an exercise. The basic concept is to chemically weave a dense spiderweb-like mesh of soluble polymer, basically the same stuff you find in baby diapers. We use sodium polyacrylate, which you add water to, and it absorbs huge amounts of water, which is how baby diapers work. What if we could weave that dense spiderweb-like mesh in between biomolecules and around biomolecules, inside cells and outside cells, make it dense enough, even enough, and process the cell or tissue just right, and if we add water, we could expand the cell or the tissue evenly. So of course, all techniques owe a debt to older techniques and discoveries, and so one of the papers that inspired our team was a paper from 1980 from a physics professor at MIT, Toyoichi Tanaka, and he studied the physics of these swallowable polymers. For example, you could take a hydrogel of sodium polyacrylate and add water, as shown in this cartoon, and the polymer threads, shown as white lines, will expand apart from each other as osmosis draws in the water. So in 1980, he and his colleagues at MIT published a paper exploring the physics of this 1,000-fold volumetric increase. And they found that the physics was well behaved. It acted like a phase transition, analogous to the kinds of changes that happen when ice becomes water or water becomes steam and so forth. Unfortunately, Tanaka passed away relatively young, so I never got to meet him in person. Now, the other interesting thing that happened around the same time, 1981, just 1 year later, was that a group found out how to install these polymer hydrogels inside biological specimens. You can't just dump the baby diaper material on top of the specimen. It'll just sit there. You have to weave that web in between and around all the biomolecules, right? So in a paper from Christine Dreyer and Peter Hausen in 1981, they created a polyacrylamide hydrogel. So polyacrylamide is sort of the uncharged cousin of polyacrylate. Polyacrylate has negative charge, which helps it expand enormously. Polyacrylamide, being uncharged, will not have that kind of large transition. But anyway, they showed that they could take preserved embryos and tadpoles and so forth, embed them in polyacrylamide hydrogels, and use that stabilization to facilitate staining and imaging. So it's fun to think about. If somebody had put 2 and 2 together almost half a century ago, maybe the talk I'm giving today could have been given back then. Okay, so the basic idea is that we want to take a cell like the one on the left, this golden neuron in this cartoon, and make it something like the cell on the right, a constellation of biomolecules hovering in space. They've been pulled apart, but their relative organization has been preserved. More precisely, 2 molecules that were touching have now been pulled apart to some minimum distance, and 2 molecules that are some large distance apart have had their gap expanded by a linear factor. Now, to make this work, we had to invent several other chemistries, shown in the following cartoons. The first thing we had to develop were anchors, or handles, that we could attach to all of the biomolecules so that we could apply force to them and pull them apart. In this cartoon, biomolecules are shown in brown, and the handles are shown in purple. We have handles that attach to proteins, DNA, RNA, and in the pipeline are ways of expanding labels or biomolecules for lipids, sugars, and other kinds of biomolecular species. Next, we have to weave that dense spider web-like mesh of polymer. We do that through a process called polymerization. So we add in monomers, shown as white spheres, and they self-assemble through a process called polymerization into long chains. When the long chain encounters one of those handles, it'll form a molecular bond. So if you think about it, the polymer can expand, the handles will convey the force to the biomolecules. We're almost there, right? But there's one last trick that we had to figure out, and that's how to soften the specimen, because it's very happy where it is. We have to loosen stuff up. We can use heat, detergents, or enzymes to loosen up the molecules, and then when we apply the water, the polymer will swell, absorbing the water, but this time, because of the softening, and because of those handles, we can pull apart the biomolecules from each other. So we announced the discovery that we could physically expand biological specimens in early 2015 in a study led by 2 then graduate students in my group, Fei Chen and Paul Tilburg. And panel B is a small piece of the mouse brain, maybe a few millimeters on a side. Panel C, the same piece of brain tissue, about a day and a half later. We've physically magnified it by 100-fold in volume, about 4.5 times in each direction. The polymer starts out quite dense, as shown in the cartoon in the upper left. The mesh size, or polymer spacing, is just 1 or 2 nanometers, smaller than many biomolecules. And when you expand, it ends up in an extended conformation, like in the lower left. Here's a little time-lapse movie. It's about a half an hour compressed to half a minute. We polymerized this piece of brain tissue earlier, and we add the water right there. And I hope you can see that this piece of brain tissue is expanding before your very eyes. So by design, the very small polymer spacing, just 1 to 2 nanometers, the anchoring, the softening, the expansion being isotropic, we thought we could isotropically expand biological specimens. So, but this is biology, we have to verify it. And so in the early years of expansion microscopy, as we call it, we spent a lot of time taking pre-expansion images with a classical super-resolution microscope, like a SIM or STORM or DNA paint technique. And then we would take post-expansion images with a regular old conventional diffraction-limited microscope, the kind that almost everybody has some kind of access to in a core facility or through other mechanisms. So in magenta or pink is the pre-expansion image, taken in this case on a SIM scope, we're looking at HEX cells stained with fluorescent antibodies against tubulin. And in green is the post-expansion image taken on a regular old confocal microscope. And so the distortion is not zero, but it's very, very small, a few percent over a typical microscope field of view. It turns out that's good enough for the vast majority of biological and medical questions people are tackling nowadays. Although, as I'll show you later, we are trying to bring the error down even smaller. What about resolution? It's good that the distortion is low, but resolution is about telling things apart from each other. But we can look at things where we have a known ground truth, like a virus or microtubules, as shown here. We know what they should look like thanks to decades of super-resolution and electron microscopy imaging. And so we can take an image of our specimen, taken on a confocal after expansion, with proteins decorated with fluorescent antibodies, and then we can deconvolve it or divide it by the known ground truth and estimate how much error we've added. And so we get an error, or a resolution in this case, effective resolution, of around 60 or 70 nanometers. Now, a typical confocal microscope might have a lateral resolution of, say, 300 nanometers. 300 divided by the expansion factor of 4.5 is about 70 nanometers. And so it raises the question, if we expand bigger, can we get even better resolution? As I'll show you later, we can. But even with 4.5-fold expansion, there are many things that we can do that are difficult and sometimes even impossible to do with earlier techniques. Here's a piece of mouse brain tissue, several millimeters on a side. I guess I'll use the mouse pointer here. You can see the cortex at the top, the hippocampus here. Each white square is zoomed in to the picture below, and the color code is shown on the left. Again, we're using fluorescent antibodies to decorate different proteins. Now, very quickly, in the lower left, we hit the resolution limit of the microscope. We see some purplish dots, right? But the structure is unclear. The right set of images is the same specimen, same microscope as on the left, but now we've expanded it. And now I hope you can see the purple dots are not purple dots. You can cleanly see blue and pink parts. And those are pre- and postsynaptic proteins at neural connections called synapses. If we look at the distance between these protein densities, we get exactly the same answer that Catherine Dulac and Zhao Zhuang measured using storm microscopy many years ago, shown in the upper right. But now we can make the same measurement on a regular confocal microscope. So expansion sort of democratizes nanoimaging. Anybody can now do nanoimaging with a conventional microscope, with off-the-shelf preservatives, stains, and cells and tissues. Now, something interesting happens when you expand in water, right? If you expand an object 100 times in volume in water, it's about 99% water. And so as a byproduct of the expansion process, it becomes completely transparent, and furthermore, refractive index matched to water within a fraction of a percent or so. What that means is you can use cool tricks like light sheet microscopy, where you shine light from one angle and take pictures on a camera at a 90-degree angle. For transparent specimens, this lets you go blazingly fast speeds because you can get a single two-dimensional sheet on each camera frame. Working with Eric Betzig's group, another super-resolution pioneer, Rei Gao, Sho Asano, and Goku Opadiula explored using expansion plus a lattice light sheet microscope, a microscope developed by Betzig in his lab. Here you can see in the upper right bits of the mouse brain where we use fluorescent antibodies to label mitochondrial proteins and lysosomal proteins in the upper right, myelination in the lower half of the slide. And using the light sheet microscope, the lattice light sheet, we were able to go about 1,000 times faster than an equal resolution classical nanoimaging method. So I'll just show you a movie here about how this increase in speed is not just about speed, it gives you scale. Here we're zoomed in, maybe too much, into a little piece of the mouse brain where fluorescent protein is in yellow and pink and blue are, again, pre- and postsynaptic proteins. And now we're going to zoom out. You can see that through expansion plus light sheet imaging, we can image from nanoscale to macroscale. In fact, we can look at the entire thickness of the mouse cortex, but not lose sight of individual synaptic connections. Eric, being at the Janelia Research Campus at the time, we have many colleagues there, of course, who focus on the fruit fly brain. And so a team working as part of this collaborative labeled all the dopamine neurons in the fruit fly brain with a fluorescent protein, and furthermore stained all the synapses with an antibody. And the synapses are not shown in this movie, but I just want to show you a fly-through of the fly brain, which emphasizes the crossing of scale from nano to macro. So here is zoomed out. Whoops. Click play here. The color codes are just made by human investigators, but now we're going to zoom all the way in, and I hope you can see individual axons and dendrites. And now we're zooming out to the lateral edge of the fly brain, and now we're going to go back out so you can see the entire fly brain. So I hope this highlights that expansion microscopy not only lets you use a regular microscope as a nanoimaging device, but if you are willing to put in the effort to use a specialized microscope, you can achieve scales that are extremely useful for answering questions that might be difficult or sometimes impossible with older strategies. Of course, these neurons are fluorescent protein expressing. Lots of people are expressing combinations of fluorescent proteins, the so-called rainbow technique, where you can use viruses to equip different neurons with random sets of fluorescent proteins, developed by Jeff Littman and colleagues. Here's a piece of the mouse brain where different neurons in the hippocampus are expressing random sets of fluorescent proteins. Let's zoom in to a small piece of an axonal region, and it's pretty blurry when you look at the confocal image. But in the upper right, you can see after expansion that the confocal can now easily resolve individual axons and dendrites. And so many groups are using some kind of color coding plus expansion to look at parts of the brain and the wiring within. So far, what I've showed you is all protein imaging, but you can look at other kinds of biomolecule as well. When Oswasi joined the team, we extended our horizons to look at nucleic acids as well. So here, we're anchoring and expanding RNAs away from each other. You can see pre-expansion, in situ hybridization against individual RNAs, and then post-expansion with the same probes on the same cells. I hope you can see that the post-expansion images for these different RNAs give you much more resolution than the pre-expansion images, and even can show you interesting conformations of those RNAs, such as the donut-shaped organization in the lower right for the long non-coding RNA NEAT1. These are in cultured cells, but you can also do this with intact tissue. Here we're expanding proteins and RNAs away from each other, and then looking at the proteins such as YFP, here shown in green, and the pink dots are individual messenger RNAs. After all, neurons have one nucleus, but thousands of connections. It makes sense that gene expression would be spatially regulated, and now we can map with nanoscale precision individual RNA molecules in intact brain tissue. Now, many groups are looking at RNAs, and in a very exciting direction, of course, is the multiplex imaging of RNAs. Bring in a library of hybridization probes, each with a barcode, dozens or even hundreds of such probes, and then you can read out the barcode through a clever chemical trick. So we started wondering, what if we could use sequencing as that clever chemical trick? And when you sequence a nucleic acid, you're basically copying it, and as you copy it, it blinks out its genetic code because you're adding fluorescent nucleotides. So sequencing, at least the way it's done most popularly, is basically imaging. So we wondered, could we take a library of probes, such as the purple padlock-shaped probe shown in the upper left, apply them to a set of RNAs, and then not shown as a little barcode on the other side of the padlock probe, which we could then sequence. And so each dot would blink out the barcode, which then would tell us what nucleic acid was found there. If we could do that with expansion, we could do multiplex imaging of many biomolecules at once with nanoscale precision in an intact cell or tissue. So in work that was quintuply spearheaded by Shahar Lon, Dan Goodwin, Ani Sinha, Oswasi, and Fei Chen, we showed that we could expand brain specimens or cells, bring in dozens or hundreds of these probes, and then look at their gene expression. And so this is quite powerful. For example, taking inspiration from Aaron Schumann's work and others, we looked at RNAs that are known to be out on delicate processes from neurons, and by expanding and doing in situ sequencing, we could find the location of individual RNAs with nanoscale precision in delicate neural architecture. And furthermore, we could find patterns. So for example, neurons have dendrites that point in different directions. We can map out the rules of how these are expressed in different directions, and even in little tiny compartments of synapses called spines, and how they're different from neighboring dendrites. So for example, dendrites that point in different directions had similar distributions of RNAs, but these spines, these nanoscale compartments, had radically different distributions than the dendrites that are just tens to hundreds of nanometers away. You can, of course, apply all this to tissues that are not brain. I'll get to this more at the end, but I'll just give you one example here. Working with Aviv Regev's group, then at the Broad Institute, she gave us a breast cancer metastasis to the liver and gave us 300 expressed genes to look at. And we did in situ expansion sequencing of this specimen, and we were able to derive interesting patterns. I'll just give you two examples. In the lower left, of course, tumor immunotherapy is a very important area in cancer right now. By looking at immune cells, such as B cells, and how close they were to tumor cells, we found that the closer the B cell, the immune cell, was to the tumor cell, certain genes would radically change their expression. And the high precision enabled by the expansion method allowed us to get very, very clean analysis of these cells. So for example, this gene S100A8 had a fourfold upregulation when the immune cell that it's in was near a tumor cell than when it was far. Hypoxia is also another interesting and rapidly expanding area within cancer. When a tumor cell was near a fibroblast, we saw a large upregulation, fivefold regulation upwards of a hypoxia-related factor. So expansion sequencing allows us to look at patterns in gene expression with great spatial precision across extended tissue specimens, such as this one from a human patient. As a side note, this doesn't involve expansion, but you could do in situ sequencing of the genome as well. We teamed up with Jason Bonrostro's group and Fei Chen's group, now at Harvard, the same Fei Chen, now running his own group. And in work that was led by Andrew Payne, Zach Chang, and Paul Reginato, we found that we could do in situ sequencing of DNA while it was still inside cells. Basically, as the cartoon at the top shows, we can knock in little barcodes all over the genome, start sequencing those barcodes, and then basically keep going, sequence the DNA of the genome next to the barcode. And so that allows us then, as you can see at the bottom of the slide, to map out not just sequences, but the 3D organization of the genome. Our hope is now to combine this with expansion microscopy so that we can incorporate greater resolution. Okay, back to expansion. So everything I showed you so far is expanded by about fourfold or roughly. But can we expand more? Well, when J.B. Chang was a postdoc in the group, we explored the following idea. What if you take a specimen and expand it, as shown in the cartoon at the top, form a second polymer in the space opened up by the first expansion, and expand it again? So it's kind of like PCR, but for space. You get exponential amplification in space with a linear number of steps. More recently, in a paper led by Deblina Sarkar, Jinyun Kang, and Oswasi, with great help from Margaret Schroeder, and Amuche Emanari and Christina Kitko have made important contributions as well, we found a way to do this iterative expansion method with all off-the-shelf chemicals, so I'll focus on that today. This is a paper on bioRxiv and just accepted at Nature Biomedical Engineering in principle. And this method illustrates a second feature of expansion, which I think is now going to become more and more appreciated. Up till now, most of us have focused on expansion as democratizing nanoimaging. But expansion has a second feature, which might be as important. In this cartoon, the top row shows proteins, shown in light blue, all clustered together. And many proteins are compacted together in complexes, right? That's how many of them work. Proteins talk to each other by binding and interacting. The red and green antibodies can decorate proteins on the outside of the complex, but they're too big to get inside. The proteins inside are effectively invisible. But if you pull the proteins apart, now the yellow and blue antibodies can access their targets. We wondered, could we take invisible biomolecules and make them visible? After all, in light microscopy, what we mostly do is to see what we've labeled, right? We looked at synapses, which are very densely packed protein agglomerates, and wondered if we could see proteins that were previously difficult or even impossible to see. This is a crowded slide, but bear with me a little bit. Panels B, C, and D showed proteins where the yellow is pre-expansion staining, and there's not much yellow. And then pink is post-expansion staining. Same fields of view, same labels, same microscope. I hope you can see there's a lot more pink than yellow. This is a calcium channel in panel B, a presynaptic protein in panel C, and a postsynaptic protein in panel D. We saw that these proteins appeared at synapses and not at nonspecific locations because not all proteins were invisible with pre-expansion staining. In panels F, G, and H, we saw similar patterns with yellow and magenta. And so we used those as reference stains to help us conclude that indeed the increased staining is at synapses and not just nonspecific. Working with Tom Blanpete's group at UMaryland, we wondered, these invisible proteins, can we look at their nanoscale architecture? And now that we saw that we could see a calcium channel subunit that was previously invisible, we started looking at how this presynaptic calcium channel correlated with postsynaptic proteins. Maybe the precision of neural signaling is governed by the nanoscale organization of proteins across the synapse. And to make a long story short, we do see within 50 nanometers a close coordination of presynaptic proteins, like this calcium channel, with postsynaptic proteins, such as PSC95, which might help with the precision of neural signaling. What about diseases? Many brain diseases and diseases throughout the body are associated with densely compacted proteins. In Alzheimer's, for example, amyloid plaques are one of the most famous hallmarks of Alzheimer's, although their precise role in Alzheimer's remains controversial. We wondered, collaborating with Li Wei Sai's group at MIT, is there something we can learn here? Well, we looked at 5X FAD mice. These are mice engineered to get Alzheimer's-like symptoms, and again stained with an antibody against A-beta 42, a component of amyloid plaques. Yellow is pre-expansion and pink is post-expansion, and we saw a lot more after expansion than before. In fact, we even saw these amyloid dots, which to our knowledge had not been described before, and which were pretty much invisible, very, very hard to see at all with pre-expansion staining. We saw this with multiple independent antibodies against A-beta 42, and we never saw them in wild-type regular mice. Because we expanded these things, we could not only detect them, we could also see what's inside. Amazingly, inside these amyloid dots, we found physiologically relevant proteins, like ion channels. Here's a potassium channel, Kv7.2. We did lots of analyses of these and tried to map out the configuration of this ion channel inside the amyloid dots. And so maybe this puts forth new hypotheses of the pathology of Alzheimer's and new roles that amyloid might play in Alzheimer's disease. Pablo Valdez-Quevedo, a neurosurgeon working with us, wondered, if we can see invisible proteins, might there not be invisible cells? He's a neurosurgeon and works with brain cancer patients. And cancer cells, of course, are densely packed full of proteins, many of which are hallmarks of different pathological processes. So in glioma patients, brain cancer patients, aggressive tumor cells have classically been marked by two stains against very densely packed proteins, the fibrillar proteins vimentin and GFAP. This is sort of a crowded slide, but the top 3 rows of images are brain cancer specimen from an actual human patient stained with antibodies against vimentin in cyan and GFAP in pink. The last 2 rows are the same specimen, same antibodies, but now restained after expansion. And importantly, this row D, the second to last row, is when we expanded, labeled, and then shrank it back down. So we're going to say, look, is the improved labeling important, even if you don't care about the resolution? Now, we wondered, if vimentin and GFAP label the aggressive tumor cells, might we see more of them if we labeled them post-expansion? We had a team of people count the putative aggressive tumor cells in a low-grade glioma patient's specimens. And if you just focus where my mouse is pointing, this is V and G, that's vimentin GFAP double-labeled cells. So again, the putative aggressive tumor cells. With pre-expansion labeling, the gray bar is the count. With post-expansion labeling, same antibodies, same specimens, we saw 6-fold more of these putative aggressive tumor cells. So what if we have been undercounting or missing entire populations of tumor cells in one of the most devastating forms of cancer that we know of? Okay, so in the take-home message of the first 2 parts of this section are expansion can help democratize nanoimaging, so almost anyone can do it, and that the crowding of molecules from each other makes them more accessible to labels, revealing previously invisible proteins and perhaps even invisible cells. Now, how precise can we expand? Can we get down to single molecules? Can we get down to fractions of a molecule? Solve the structures of molecules, for example? Well, all the methods of polymerization I've told you so far are using free radical polymerization, which is sort of very related to that paper I mentioned from 1981. But polymer chemistry is a rapidly growing field, and many people are inventing new kinds of polymers, such as this diamond lattice polymer shown in the cartoon at the right. We wondered, if we use these new polymers, which might have interesting properties, could they expand more evenly? Now, this diamond lattice polymer is thought by polymer chemists to be nearly defect-free. In contrast, the free radical polymers that expansion has used to date have defects as shown in the upper left: denser areas, dangling ends, and loops. In contrast, the diamond lattice polymer, which is made out of these tetrahedral monomers that grow end to end, are thought of as nearly defect-free, thanks to methods of imaging polymers like small angle neutron scattering and so forth. So Ray Gao and Jay Yu in our group wondered, could we do more even expansion by adopting new ideas from polymer chemistry in recent years? Well, we collaborated with Rachel Neve's group at Harvard to expand virus particles. And we were just sort of thinking of viruses as sort of nature's scale bar. And so we used HSV-1 viruses from her lab. The upper left shows these HSV-1 viruses with their coat decorated with fluorescent molecules, and then expanded with the tetrahedral or diamond lattice gel in the upper left. The free radical gel also expands, and you can still see the round shape, but there's more error being introduced into the envelope of the virus. The population data is shown here. With the free radical gel, maybe we're getting 15 to 20 nanometers of distortion. But with the tetragel, we're getting 9 nanometers of distortion. Single-digit nanometer precision imaging on a conventional microscope. Zhao Zhuang's group later went on to expand DNA origami and do storm imaging, and they had a recent paper where with our tetrahedral hydrogel, they're now getting down to about 5 nanometers of precision. So I think the race is on to get down to single molecule precision with expansion, which is going to be a lot of fun. But independent of the precision, the expansion is biologically relevant as well. Here we're showing that we can just look at the shapes of these viruses with more accuracy, look at things like ellipticity and sphericity and so forth, and we can get more accurate images. I think there will be a lot of uses of this in not just biology, but also perhaps in medicine, as I've alluded to. Yongshen Zhao and Ativia Bucure, working with my group, Astrid Wein's group at Harvard, Andy Beck's group then at Harvard, showed that they could expand a wide variety of human tissue types: prostate, lung, breast, pancreas. On the left are normal, and on the right are cancer-containing specimens for human patients. The color code is in the upper right. And I hope you can see the post-expansion images show much more detail than the pre-expansion images. Working with Andy Beck, we wondered, could you detect cancer earlier if you expanded? After all, early in a disease, the changes are quite small, right? So we expanded breast cancer biopsies from actual human patients, had a committee grade them with pre-expansion images and post-expansion images, and the votes of the committee were then used to train a machine learning algorithm to classify them. And I hope you can see here, the green circles is the consistency of the classifier on post-expansion images, and the pink squares are for pre-expansion. And the algorithm was able to be much more consistent with post-expansion images than pre-expansion images. In fact, it did well even in some conditions where early in a cancer, doctors can disagree up to half the time. All right, let's wrap up the first half of the talk. We've discovered that we can physically expand biological specimens. It's helping democratize nanoimaging, and the decrowding is able to make invisible molecules more labelable and thus visible. The number of experimental papers and preprints, not like review papers, actual experimental papers and preprints, is on track to hit 400 any day now. I would estimate well over 1,000 groups are doing some kind of expansion process. And I guess the font's not quite big enough to be read on the screen, but people are applying it to all sorts of stuff. You know, the worm C. elegans, human kidney specimens, motor proteins, the nucleus, stress granules, and phase-separated compartments. And the list goes on and on and on. Pre-COVID, we hosted lots of people at hands-on workshops at Janelia, at the Salk Institute, at the Jean Paul Institute, and all over. But of course, with COVID, that had to end. In summer of 2020, we posted a photographic step-by-step tutorial on how to do expansion. It includes really, really simple steps being explained through pictures, including how do you use a pair of forceps, or how do you use a paintbrush. So we're continuing to try to teach usage of expansion. Our website, expansionmicroscopy.org, has a lot of references of protocols, including this one. And so please give it a try if you want to look at nano aspects of cells and tissues. After all, biology, I think, in the end, is about nanoscale things, right? Genes, gene products, and biomolecules are all nanoscale, and they interact over nanoscale distances. And so it's quite possible that every branch of biology and medicine could benefit potentially from a way of nanoimaging that's easy to apply. So that ends the first half of the talk. In the last half, I want to tell you a few short stories about time, because, of course, the obvious limitation of expansion microscopy is that it's never going to work, as far as I can tell anyway, on a fully living thing. And so the time dimension, of course, has been eliminated. So in the last half of the talk, I want to tell you a few short stories about time with a focus on more recent work. And I'll just mention optogenetics briefly. It's a fairly old topic, so I won't spend a lot of time on it. But I feel like I should just maybe mention some of the recent things. Optogenetics is more specific to neuroscience, so maybe I won't spend a huge amount of time on it for this broader audience. But the basic idea is to control brain cell electrical activity using light. This started when my co-inventor Carl Dijsselroth and I were both students at Stanford in the early 2000s. And we wondered, could we deliver some kind of energy to the brain and have those brain cells convert the energy into electrical activity, since brain cells, of course, compute using electrical activity. So we just started making a list of all the laws of physics. You could use magnetic fields, you could use mechanical force, you could use light. And we thought light would be very cool if you could get away with it, because it can be aimed, of course, very precisely. And of course, it's faster than anything else. You can bring light into the brain doesn't feel pain, so you can bring in an optical fiber or a small microscope or another light relaying device. The next question then, of course, is how do you make brain cells sense light? Because, of course, most of them don't. And so I became really fascinated by a class of molecule known as the microbial opsin. These are 7 transmembrane proteins that look like the lower left. They sit in the membranes of single-celled microbes. A vitamin A relative called all-trans-retinal absorbs light from the sun and kicks off a protein conformational change sequence that'll pump an ion from one side of the membrane to the other. The first to be found was a light-driven proton pump from a single-celled archaea species, and it's used by this microbe to convert light into a proton gradient, which in turn drives ATP production. Later, people found light-driven chloride pumps that also store solar energy in chemical form. And then finally, light-driven ion channels were found around the turn of the century. Flashing forward to the current day, what we found is that all of these classes of molecule contain members that are fast enough, safe enough, and powerful enough that they can be used to turn brain cell activity on or off. So when Brian Chow and Xue Han worked with me, we found that certain light-driven proton pumps could be genetically expressed, just deliver the gene with a gene therapy vector. And amazingly, the neuron, the brain cell, can make this light-sensitive protein. And it's safe enough, fast enough, and powerful enough so that when you shine light of the right color, green or yellow light, the neurons will pump out the positive charge and turn off. So by deleting neural activity, you can find out whether a brain cell or set of brain cells is needed for an emotion or a memory, or whether deleting the activity is enough to cancel out a pathological state. Xue and later Amy Chuang looked at light-driven chloride pumps, which similarly will, in this case, pump negative charge into brain cells and then turn them off. Carl Dijsselroth and I originally collaborated to put channelrhodopsin 2, a light-driven ion channel, into brain cells. We found that shining blue light on them could activate their electrical activity. So by activating neurons, you can figure out if they can trigger a behavior or a pathological state. And later, Nathan Klepetke worked on a red light-sensitive version. Red light, of course, goes deeper in the brain or body than other colors of visible light. So most of the molecules that are in widespread use these days are still molecules that we or others published in 2010 or earlier. It spread very, very quickly in neuroscience. maybe I'll just emphasize a few recent developments, which are on the application side. Thousands and thousands of people have been using optogenetics to study the brains of small animals like mice. But last summer, a paper led by Bolton Roska and José Alonso came out where they showed that you could actually express a molecule in the eye of a blind person to help this person see again. So many people have lost the photoreceptors in their eye. Their eyes can no longer capture light. What they did in this French-Swiss collaboration involving a company, Gensight, is they put a dose of crimson, the red light sensitive molecule that we published in 2014. They gave this person a single viral dose that encoded for that gene into one eye. And this person was able to look at household objects, to see the lines of a crosswalk in the road, and to have partial restoration of functional vision. It's not perfect. He couldn't read text, for example, but it's, to my knowledge, the best restoration of functional vision for these photoreceptor loss classes of diseases that has been seen to date. Another recent development is trying to get down to single-cell resolution. Collaborating with Valentina Miliani's group at the Institut de la Vision in Paris, or Shemesh and members of her group worked together to make molecules that are optimized for the holographic multiphoton stimulators that her group develops. So basically project a hologram into the brain, which is basically a 3D sculpture of light. And then what we did was to make molecules optimized for such holographic projectors. And what we found was that we could do very precise, temporally precise, single-cell control of neurons. So this area, optogenetics, opto for light, and genetics because these are genetically encoded reagents, has really taken off. And so maybe what I'd like to do is to pivot to some newer stuff for the remainder of the talk. But I just want to emphasize how lucky we were, right? These molecules existed in the natural world, pretty much fresh out of the box without modification. They were safe enough, fast enough, and efficacious enough to immediate precision control of neural electrical activity, even in, as you see, maybe human patients as well. And so I want to spend the last several minutes of the talk going into some newer work we're doing, which is about the inverse, of course, of optogenetics, which is seeing living things in action. And of course, many people over the last several decades have made genetically encoded tools that light up when a cell is active in some way, or a signaling cascade is active in some way. Take a fluorescent protein and mutate it, or take a fluorescent protein and fuse it to a sensor domain, and you can make a genetically encoded indicator of a cellular function or molecular function. So this is still a very hard thing to do. It can take an entire PhD studentship to make one molecule. And that's because protein design is still very, very difficult. So how can we be more lucky, right? With optogenetics, we were really lucky. Can we be luckier in the case of readout of cellular signaling or molecular signaling? Well, when Erika Jung and Kirill Pyakovich were postdocs in the group, we thought, let's build a robot that will do evolution. If the world won't evolve these molecules in nature, why don't we evolve these things in the lab? And so directed evolution has a long and storied history, but we wanted to adapt it, first of all, to work in mammalian cells, where the stuff we make could be used to image things in human cells, for example. And secondly, we wanted to do a multidimensional screen. With optogenetics, we got multidimensionally lucky, right? The molecules were safe, they were fast, they were strong. If we want to evolve things in the lab for safety, we might de-evolve them away from speed. If we want to evolve things towards speed, we might de-evolve them away from safety, right? In evolution, you get what you select for. So we wanted to do multidimensional directed evolution in mammalian cells. Now, what does that mean? Well, in this cartoon, it illustrates the technique we developed. Take a gene and make millions of mutants. For any given goal, like making a fluorescent sensor, some of those mutants will be better for your goal, and some will be worse, right? Just by chance. Express them, one copy per mammalian cell in a dish. Then we can scan them with a microscope, and microscopes, of course, are great for multidimensional assays, right? We can look at speed, we can look at brightness, we can look at cell health. Then we bring in a robotic arm, and this is commercially available, so anybody can do this, and suck out the cells, and therefore the genes that are better. This is what it looks like. We have an automated microscope, and then an automated suction pipette attached to a robotic arm, which can pick out individual cells and put them into wells for later genetic analysis. So we did what might have been at the time, who knows, maybe it still is, the largest directed evolution mammalian cell-based screen, 10 million variants in 2 rounds of directed evolution. And we wanted to make a fluorescent voltage indicator, sort of the opposite of optogenetics, if you will. We started with Quasar 2, a prototype from Adam Cohen's group that's very, very fast, but not particularly bright. And we wanted to evolve them to be brighter and safer and better expressed on the membrane. So this is brightness on the y-axis, and then location on the membrane on the x-axis. And we want each cell is represented by a circle. We want to pick out the cells and therefore the mutants that are along the diagonal, right? They're better in brightness and they're better in membrane localization. This is a crowded slide, but the take-home message is that we got what we wanted. We made a molecule we call Archon. It's very well expressed in the membrane and safe. It has very high signal to noise, and the kinetic properties are quite good as well. They're still fast and photostable. Working with Shuahan's group at BU, we looked at voltage in the brains of awake behaving mice. So here's a simple microscope, just shine a red laser on the brain, collect the far red and infrared fluorescence onto a camera, and what you see here look like electrical traces in the middle of the slide from different brain regions of an awake but head-fixed mouse. But they're not electrical traces. They're being imaged on a fast camera. And what that means, of course, is you can look at many cells at once, right? So here we are peering into the hippocampus of an awake behaving mouse, and we can see about a dozen cells, of which 8 were active during this period of time that we imaged. And importantly, the fact that this is red light excited means it's compatible with blue light driven optogenetics. So here we have a couple cells, again, in an awake behaving mouse brain. We're using red light to image the voltage of the neurons expressing Archon, and these blue bars are periods of time when we shine blue light on the brain and drove the neurons. So it's all optical interfacing to the brain. As a side note, this is speculative, but it's interesting to think that, wait, if optogenetic control of neurons is now being explored for human therapy, and by the way, Archon and Quasar 2 are mutations of older optogenetic tools, so they might have a similar profile of safety in brain cells in the human, potentially. Could you really build a non-contact, all-optical human brain interface? So this is clearly a direction for the future, but I think that article in Nature Medicine last summer showing that Crimson, our red light sensitive activator, could work at least in one patient, and now I think that many more patients are in the pipeline, with many companies springing up to pursue optogenetics in people all of a sudden. An era of all optical brain interfacing for human patients might be extremely interesting to contemplate. By the way, we're applying this robot to many other kinds of molecular optimizations. We're collaborating with many people, and now that travel's freeing up a bit, we're eager to welcome people to come and do screens with us if they want. And so hopefully we can help optimize many molecules for different functions. Now, of course, we're not the first people to make fluorescent indicators that are genetically encoded. As I mentioned earlier, hundreds of molecules have been made where somebody takes a fluorescent protein, let's say, and fuses it to some kind of sensor domain, and now you can measure a cellular or molecular signal. So this is a screenshot from Xinjiang's database of fluorescent indicators that are genetically encoded. People have made fluorescent indicators of calcium and cyclic AMP and protein kinase A, and this goes on and on and on. And so we're making some sensors too. Cristina Torres-Caban just had a paper a couple weeks ago where we made a prototype fluorescent potassium indicator. But it raises the question, how do we use these indicators to really understand an entire system? After all, the genome encodes for thousands and thousands of different biomolecules, right? A given cell might take dozens of different inputs or more and make dozens of different outputs, and inside is a sea of interaction. How does a cell take in inputs, compute what it needs to do, and then generate the right outputs? Well, wouldn't it be great if we could see many signals at the same time? And that's important to see the relationship between inputs and computation and outputs. I'll just give an example, right? Suppose when input A is high, computational signal B is low. When signal A is low, signal B is high. If you image A in one set of cells and B in a second set of cells, you might miss out on that important relationship, right? Ideally, you could see A and B at the same time, and then the relationship would just jump out at you. Now, the classical way of doing that is to make different indicators have different colors. But that means if you want to make an indicator a new color, you have to make it, basically from scratch, right? And that can take a long period of time. Furthermore, what if you want to see more than a couple things? What if you want to see 3 or 4 or 5 or even more signals at the same time? Well, Shannon Johnson and Cheng-Yang Ling-Gu in my group worked on a totally different way of exploring this possibility, shown in the cartoon in the upper left here. What if you take different fluorescent indicators, even ones that are off the shelf, made by other groups and documented in Jin Zhang's database, right? What if you park one set of indicators, let's say for calcium, at the points labeled 1 inside a living cell? And suppose you could park a second indicator, I don't know, for protein kinase A, at a second set of points, the points labeled 2, in the same living cell. So while the cell's alive, each of those points will be a little dot of indicator blinking at a little movie of what's going on, right? Even if they're all the same color, like green, each dot could tell you a different story. In theory, you could have hundreds of indicators. A cell is, of course, fairly large for the most part compared to the resolution limit of a conventional microscope. Now, of course, sometime you have to figure out which signal was at each dot. So at the end of the experiment, you can preserve it and then use multiplex strategies like the kind that I described in the first half of the talk to figure out which sensor was at each point. Let me give you an example of how we can do that. So there's several ways that we figured out you could do this, but our favorite way is to take different fluorescent reporters and fuse them to different self-assembling peptides. Those will safely and stably cluster at different points in a cell. Self-assembling peptides have been designed by protein engineers for many years, mostly just to explore the science of protein engineering, but here we have found a practical application of them. So suppose you have one reporter fused, let's say for calcium, fused to one self-assembling peptide. It'll cluster in little points as they self-agglomerate. A second reporter is fused to a second self-assembling peptide. It'll agglomerate at a different point. And by the way, each of these reporters has a different epitope. An epitope is just something we can stain later once we preserve the specimen. So each epitope is associated with a different antibody. And so we can bring in fluorescent antibodies at the end and label the epitopes for imaging, potentially over many rounds of staining and imaging. So here's a little cartoon of what that might look like. The live cell image would be a bunch of green dots, let's say, blinking at little movies of the signals there found. Then you can preserve the cell and stain it potentially over many rounds of staining and imaging and reconstruct the false color image here shown. Well, it works. So in panel B is the much-loved calcium indicator, GCAMP6. We expressed it in a cultured neuron. Panel C, the same calcium indicator fused to a pair of self-assembling peptides. We use a pair to make the cluster somewhat bigger. And you can see that it forms puncta. We did lots and lots and lots of experiments to look at the safety of the cell, to look at the quality of the signal, to look at the nature of the cell's physiology. Supplementary figure 1 of this paper has 26 panels of control experiments, and the list goes on. And we didn't see any changes in cell health, cell physiology, or sensor quality. So the moment of truth. Can we take 3 different off-the-shelf fluorescent indicators, fuse them to different self-assembling peptides, and get them to park at 3 different sets of locations in a cell? Well, we took indicators for calcium, GCAMP6, cyclic AMP, CAMP-R, and protein kinase A, X-ray ACAR1, and expressed them in the same cultured mouse neuron. Green would be what the live images look like, and the false color images are when you're done staining and imaging. And look, you can see 3 different signals in the same living cell, and if you drive it with forskolin, which drives cyclic AMP, you can see calcium, cyclic AMP, and pKa signals. So to wrap up, what you can see is the relationship between different signals. Some cells, like in H, had slow calcium signals when forskolin was driven. Some, like in I, had fast calcium signals. We wondered, calcium is sort of one of the inputs to this molecular circuit. Can we predict the outcome, which is the sustained pKa activity? Well, the fast calcium neurons had strong pKa output, and the slow calcium neurons had weak pKa output. so we can see the relationship between the timing of one signal and the amplitude of another. So we're very excited with the prospect now of imaging a wide variety of different signals in the same cell to see their relationships. To summarize the last part of the talk, we've been trying to be lucky, whereas with optogenetics we just were lucky. And we're trying to be lucky both through robotic strategies to make indicators better, as well as developing new strategies for using existing indicators in powerful new ways. I think of acknowledge along the way all the people who led the projects, but I'll just end on this slide with an even longer list of people at the top who helped with these projects in the group over the years, and an even longer list of groups all over the world who we've worked with to develop and apply these tools. Please visit our website, synthneuro.org, because we share all of our tools as freely as possible. And in addition to being the PI of the group, I'm also the chief tech support person, so don't hesitate to email me. It's just my name, Ed Boyden at MIT.edu, if we can be helpful. And with that, I can take questions.
Thank you so much, Ed, for such a wonderful and inspiring talk. So unfortunately, we're very tight on schedule, but we will have time for one quick question from the audience. There's a question over there. Yeah.
Hello. Thank you. That was wonderful. I just had a perhaps conceptual question. So always my interpretation in my brain was that these more messy polymers that you use for expansion, one of the key aspects was that you had this sort of wobbling empty space that could get then elongated. But then you show this strategy of a much more organized polymer, but then still you managed to expand it. So I think something that I was missing. How can you manage to expand it if it's so well organized and it's such a symmetric gauge?
So the tetrahedral monomers that form the diamond lattice, the picture I showed is in the extended conformation. They start up much more compact because we add salt, which allows, through charge screening, the collapse of the monomers to become smaller. That could mean that because we are attaching the monomers when they are in a bit more of a disordered state, that it's not completely defect-free. As I mentioned, our resolution was not perfect. We had 9 nanometers of error. But we have lots of ideas now about how to try to improve that further. Yeah, great question.
OK, fantastic. We still have time for one more question. Thank you.
Thank you. It's going to be really quick. On the part where you're talking about expansion microscopy, and you're showing it in the single-cell level, before and after, I didn't see a change in the z-scale, whereas I thought it was going to be isotropic, and it was going to expand in XYZ, but then the z-scale bar that was there was for the same height of like 5 microns. So I was wondering if the cell had been constrained
No.
its z-axis?
No, not at all. So just to make the images easier to see by eye, when we take a certain number of z-stack slices in the pre-expansion state, we then take a larger number of z-stack slices to tile the same volume, biologically speaking, in the post-expansion state. Otherwise, the images would look quite different. But the expansion is isotropic in all 3 directions. Yeah, great question.
All right, thank you very much. So I think we have to move on with the program. So please join me in thanking Ed for his amazing talk. So as you can see, there's been a bit of background activities on the stage while we were taking the question and answer. But this is actually now the end of the scientific part of the meeting for today. So I would like to thank you, all of you, for contributing to the discussions, and all the speakers for contributing, of course, to the scientific content of the talk.